Welcome back to the deep dive. If you're tracking the cutting edge of oncology, well, you know that advanced prostate cancer research is moving incredibly fast.
It really is.
So today, we're taking a comprehensive look at the very newest strategies. We'll jump straight into the data from some really innovative clinical trials and also some truly revolutionary preclinical work that's shaping the field.
Yeah, and what really stands out when you look at these sources, it's the absolute commitment now to precision and synergy. The research focus has fundamentally shifted, you know?
Oh, so?
Well, it's not just about hitting one target hard anymore. It's about convergence. It's about creating these highly targeted, often dual-action therapies that attack the tumor's weak spots on multiple fronts.
Okay, so hitting things like hormonal signaling, metabolism, immunity.
Exactly. Even the delivery tech itself, it's getting really sophisticated.
Right. So that's our mission today, to kind of distill these complex updates, everything from phase 3 trials right through to machine-driven discovery. We want to give you a complete up-to-the-minute view of where the science is actually headed.
And we figured the best way is to start with the research that's, impacting patients right now in the clinic.
The clinical trials.
Before we dive into the foundational lab work, which is really showing us the immediate future of treatment.
Okay, so let's kick off with a big one. A phase 3 trial that really shows this dual strategy. This is in metastatic castration-resistant prostate cancer, mCRPC.
Right.
It's combining Xaluritamig, that's AMG 509, with the established hormonal agent Abiraterone acetate.
Yeah, this combo really tries to tackle two key problems at once. Abiraterone, well, we know it inhibits androgen synthesis, essentially starving the cancer hormonally.
Standard approach.
Pretty standard now, yeah. But Xaluritamig, that's the really novel part here. It's an advanced bispecific T-cell engager.
Okay, break that down. A bispecific T-cell engager.
Think of it like a molecular matchmaker. It has two arms. One grabs onto the immune system CD, 3 receptors on T cells.
Right.
The other arm grabs onto the STEAP1 protein. And STEAP1 is expressed on, get this, over 95% of prostate tumors.
Wow, 95%. So you're using Abiraterone to slow the cancer growth hormonally. And at the same time, Xaluritamig is basically painting a huge target on the tumor and telling the body's own T cells, go get them.
Precisely. It drags the T cells right to the cancer cell.
That sounds incredibly powerful. But what's the potential downside? Is toxicity a big worry with these bispecifics, or does targeting STEAP1 help keep that focused?
That's always the critical question with immunotherapy, isn't it? The hope is that because STEAP1 is so specific to the prostate cancer cells, it localizes that immune attack mainly to the tumor.
Minimizing collateral damage.
Exactly. And the results so far suggest this combo is very effective at delaying progression, especially in patients who haven't had chemo yet. It's a really smart mix of targeted immune activation and standard hormonal suppression.
Okay, that idea of localized, really powerful killing, that connects directly to the next thing, next-gen radioligand therapy. We've got updates from the phase 1 ARTISAN trial. They're looking at a totally new approach using an alpha radioligand therapy, AB001.
Right. So most people might have heard of lutetium-177 PSMA therapy. That uses beta emitters.
Yeah, that's become more common.
Well, this new one, AB001, it's potentially a big leap forward because it uses an alpha-emitting isotope, lead-212.
Alpha emitter versus beta emitter. What's the difference in practice?
It's substantial. Think of beta emitters like maybe shotguns. They're useful, but the effect spreads out a bit. Alpha emitters, they're like microscopic guided missiles. They deliver highly localized, very high energy radiation.
Because the alpha particle itself doesn't travel far in tissue.
Exactly right. It dumps its massive energy payload right at the tumor cell it hits, which should mean much less damage to nearby healthy tissues. So AB001 links a PSMA targeting molecule to this lead-212 isotope.
And that isotope has a fairly short half-life.
10.6 hours.
Right.
Which is good for delivery and clearance. And crucially, the ARTISAN trial is looking at this in patients who've never had lutetium therapy and in patients who've already progressed on it.
Oh, that's important. Addressing that resistance issue.
Yes, a major unmet clinical need right there.
Okay, so we've covered immune approaches, radiation, but what about when the cancer just gets resistant to standard hormone therapy itself? We need a different kind of combination then, right?
Absolutely. And that leads us nicely to a phase 1-2 study combining Tulmimetostat and JSB 462.
Okay, 2 more new names.
Yeah, this is a really sophisticated attack on how the cells resist treatment. First, you have Tulmimetostat. It blocks 2 enzymes, EZH2 and EZH1.
EZH2 and 1, what do they do?
You can think of them as part of the cancer cell's stealth and repair kit. They help silence tumor suppressor genes and let DNA repair run wild. Locking them basically disables the cancer's repair shop and makes it more vulnerable.
Got it. So that's one punch. What's the second agent? JSB462.
JSB462 uses one of the most exciting new technologies out there, PROTACs.
PROTACs. We are hearing that term a lot lately. How does this one work?
So a PROTAC, that stands for proteolysis targeting chimera, it's fundamentally different from a standard blocker drug like, say, enzalutamide.
Okay.
Instead of just sitting in the receptor trying to block it, the PROTAC acts more like a biological garbage disposal truck. It actually hijacks the cell's own machinery to chew up and degrade the entire androgen receptor protein.
Wow, so you're not just blocking the signal, you're actually telling the cell to destroy the receptor itself?
Exactly. You eliminate the driver. This is potentially huge for overcoming the many ways mCRPC learns to get around standard hormone therapies that just try to block the receptor. It's a really cool mechanism.
That is clever. But all this focus on efficacy, hitting the cancer harder, it always needs to be balanced against the patient's actual experience, their quality of life.
Couldn't agree more. And that's precisely what the next trial we looked at addresses. Personalized dosing. It's a phase 2 trial exploring something called PSA-Triggered intermittent therapy. This is for metastatic hormone-sensitive prostate cancer, mHSPC.
Hormone-sensitive, so earlier stage disease.
Right. And the context here is simple. Continuous therapy like ADT plus an advanced androgen receptor pathway inhibitor, and ARPI, it works, but the side effects can be really tough. We're talking serious fatigue, metabolic problems, cardiovascular risks.
Things that really impact daily life.
Hugely. So in this study, patients start on continuous therapy, say Relugolix, plus an ARPI, until their PSA level drops down to a very low point, a nadir.
Okay, and then what? They stop?
Exactly. They get a break. They pause treatment completely.
Yeah.
They only restart if and when their PSA starts to climb back up past a certain predefined threshold.
So you're using PSA level as a dynamic trigger tailored to each patient.
Precisely. It's individualized. The goal is to maximize the time off therapy, reduce the side effects, improve quality of life, hopefully without losing overall control of the cancer. It's about finding that balance.
That makes a lot of sense. Okay, before we move into the future, the preclinical stuff, there's one more piece from the clinic that's crucial. Diagnostics. Better imaging helps make all these targeted therapies work better, right?
Absolutely critical. You need to see the target.
So let's look at the results from the phase 2 Co-PSMA trial. This compared a new copper-based PD imaging agent, 64 copper SAR-based PSMA, against the standard gallium-based one we use now.
And the results were pretty clear. The copper agent came out on top. It detected a statistically significantly greater number of lesions per patient.
Why? What's special about the copper?
It really comes down to the physics of the isotope, copper-64. It has a much longer half-life than gallium-68, about 12.7 hours versus just over 1 hour for gallium.
12 hours versus 1 hour. That longer time window must allow for something different, like delayed imaging.
Exactly. That's the key advantage. You can image the patient later, sometimes even the next day, after the tracer has had more time to circulate, clear from the background, and really accumulate in the tumors.
And does that make a difference?
A big difference. They found that next-day imaging with the copper agent revealed almost double the number of lesions compared to same-day imaging or the standard gallium agent. The contrast is better.
Double the lesions detected.
Yeah, and it allows clinicians to spot lesions as small as maybe 2 millimeters. That can completely change how you stage the cancer or how you detect recurrence much earlier. It's a big step up in precision.
Okay, that's a great overview of what's happening in trials now. Let's shift gears, look into the crystal ball a bit, the groundbreaking science happening in the lab. And interestingly, that same theme of synergy, of dual targeting, it really dominates here too.
It absolutely does.
Let's start with what some researchers are calling the guardian enzymes of the androgen receptor.
Yeah, this is really compelling preclinical work. They identified these 2 specific enzymes. They're protein disulfide isomerases called PDIS1 and PDIA5.
PDIA1 and 5.
And what they found is these enzymes act like molecular bodyguards for the androgen receptor, the AR, which is so critical in prostate cancer. They help keep the AR stable and functional.
So the idea is if you can knock out these guardians,
then the AR itself becomes unstable and the cell degrades it. You take away the cancer's main driver signal.
Okay. That makes sense as a target. But the researchers found something more, right? A kind of secondary effect.
They did, and this is where the synergy comes in. Inhibiting PDIA1 and 5 does more than just destabilize the receptor. It seems to directly attack the cancer cell's energy production by causing severe mitochondrial dysfunction.
Hitting the power source.
Exactly. And it ramps up oxidative stress within the cell, so you're kind of cutting off the fuel and causing internal chaos at the same time. But the really foundational finding was the synergy. When they combined these enzyme inhibitors with enzalutamide, the standard AR blocker.
The results were far, far better than using enzalutamide alone.
Yeah.
You're hitting the signaling and the energy supply simultaneously.
That multi-hit approach again. It really seems to be the theme.
Definitely. Yeah.
Okay, so that kind of powerful synergy. It brings up the next big challenge, which is always delivery, right? We can design these amazing molecules, PROTACs, inhibitors, whatever. But getting them where they need to go in the body, stably and safely, that's tough.
Incredibly difficult, a major hurdle.
Which leads us to the development of these things called in vivo self-assembled Nano-PROTACs. Sounds complex.
It is complex, but it's designed to solve a real problem. Standard PROTACs often have, let's say, poor pharmacokinetics. They might not get into cells well. They can be unstable in the bloodstream. There's something called the hook effect, which limits their effectiveness.
Okay.
So this new system designs the PROTAC component so they actually spontaneously assemble themselves into stable nanostructures once they're inside the body.
They build themselves in vivo. Yeah. And these nanostructures seem to circulate much better and penetrate tumors more effectively.
And is there an advantage to this nanostructure beyond just stability?
Yes, a crucial one. The design allows for precise, localized activation. The therapeutic part, the bit that actually triggers the degradation, is designed to only become fully active within the specific, slightly acidic environment of the tumor.
So it's less likely to cause problems elsewhere in the body.
Exactly. It minimizes systemic toxicity, protects healthy cells. It's a potential solution for translating these really potent molecules into safer, effective drugs.
Very clever engineering. Now, we also saw preclinical work targeting some really specific, tough subtypes of resistant prostate cancer, the ones with PTN loss and PI3K activation. That's quite common, isn't it?
It is. Maybe around 40%, give or take, of advanced cases have this specific molecular signature. It makes them very hard to treat.
So what's the new strategy here?
This was a combination therapy targeting two pathways simultaneously, MCL1 and AKT. They used a drug called fadraciclib, which indirectly lowers levels of MCL, one that's a protein that stops cells from undergoing programmed cell death, apoptosis.
So you want less MCL1.
Right. And they paired that with an AKT inhibitor, either epotassertib or capivasertib. AKT is a major survival pathway, especially when PTN is lost.
Okay, hitting two pro-survival signals. And did it work?
Well, here's the striking part, and it speaks to that specificity again. When they use these drugs as single agents in their models, virtually no effect on tumor growth.
Nothing.
Almost nothing. But when they combined them, they saw a significant slowing of tumor growth and increased cancer cell death. It really hammered home that for certain resistant subtypes, you absolutely need that precise two-pronged attack to overcome the cancer's built-in survival mechanisms.
Fascinating. A combo that only works for a specific genetic subtype.
That's the direction things are moving.
Okay, finally, we absolutely have to talk about the AI revolution hitting drug discovery. This collaboration between Yale and Google DeepMind on the Cell 2-sentence scale 27B AI model, this feels different.
Oh, it is. This isn't just AI crunching existing data faster. This is AI generating novel, testable therapeutic hypotheses, seemingly independently.
So what did the AI predict in this case?
It analyzed just vast amounts of cell data, cellular function data, and it proposed a completely new synergistic mechanism. It predicted that a drug called cilmetasertib, it's a CK2 kinase inhibitor, would enhance something called antigen presentation.
Making the tumor cells more visible to the immune system.
Exactly. But here's the kicker. The AI predicted this would only happen effectively if cilmetasertib was combined with very low levels of interferon, a very specific context.
Wow. Okay, that's a specific prediction. Did they test it?
They did. The Yale researchers took that prediction and tested it in human neuroendocrine tumor cells. Crucially, this was data the AI model had never seen before. And the model was right. They observed about a 50% increase in immune visibility markers when the two were combined at the predicted low interferon levels.
That's incredible. The AI came up with a mechanism humans hadn't, and it was validated experimentally.
Yes. And that's why it's such a potential landmark. It's thought to be the first therapeutic mechanism proposed entirely independently by an AI and then proven correct in the lab. It suggests these huge language models trained on biological data can almost function like virtual cells.
Wow.
They can reason through incredibly complex, context-dependent drug interactions that might just escape human researchers looking at the same data.
That is genuinely mind-blowing. It makes you wonder if AI can operate like that, like a virtual cell, proposing these complex pathways. What are the next big barriers in translational medicine, in getting drugs from the lab to patients, that this kind of technology is going to break down? And I mean, how much faster could the pace of discovery actually get across all of oncology? It's quite something to think about.
It really is. The potential is enormous.
Stay strong and fight on.